Welcome to MOOC course on Introduction to Proteogenomics. So, we are proceeding from the protein quantification, running on the SDS page gel, doing the sample cleanup, peptide quantification. Now, you are ready to inject the sample in the LC liquid chromatography followed by the MS analysis. So, now it comes the liquid chromatography. Here we are using reverse phase column C18 material. Peptides are going to bind to the column and then you want to elute the peptide based on the hydrophobic hydrophilic properties. So, you will use a different gradient using acetonitrile 5% to 80% or you can go even up to 90% with 0.1% formic acid. And this concepts I have already briefed in the beginning in the previous lecture. So, I am not repeating again, but you need to pay attention to the parameters for what should be the best gradient for doing the liquid chromatography. So, what is shown on the screen here is again a refresher we talked earlier as well, that you will use different parameters depending on the kind of sample you have, and you would like to achieve a good Gaussian distribution of the peptides which are eluted from the column. You would like to see that very soon, maybe after 5 to 10 minutes time, peptides start eluting out of the column. Then after, as you increase the gradient of acetonitrile, more and more peptides are coming out of the column, and eventually once you have reached to the saturation level, then finally all the peptides are out of the column and you are then washing and recalibrating it, being ready for the next injection. So, that ideal setup should give you a Gaussian distribution of good intensity of all the peptides. One need to play with these parameters, but again shown here that you need to work on each of these, and now we will talk more about these parameters in the lab session.
Hello. So, here I am going to explain about mass spectrometry. So, basically there are main two components, one is liquid chromatography, another is mass spectrometry. So, I am going to explain first about liquid chromatography. So, here you can see there are main two solvents, which is one solvent A which having a 0.1% formic acid, another is for solvent B which is 80% ACN in 0.1% formic acid. Now, here if you see this screen, now here you can see there are pump A and this is pump B, basically which regulate the flow of solvent A and then solvent B. This is another pump which is pump S. It control the taking of the sample, that how much amount of the sample has to be injected, that will be controlled by pump S. So, here I am ejecting the tray where you can keep your sample. Now, here you can see this plate, okay, and this is vial where you can keep your sample. So, generally here we are loading around 1 microgram. Accordingly, you can calculate the amount of volume how much you have to inject in mass spectrometry.
Now, you are familiar with the liquid chromatography and how nano LC can be used, how different parameters, different pumps are important, keeping a watch on the pressure is very crucial. Now, you are ready for injection with the electrospray ionization. This is the very crucial part, because now all your peptides have converted to the gaseous ionized forms and they have to move it inside the mass analyzers for further analysis. So, various settings and again the voltage, these criteria are important, and you are going to learn more about them in the lab session, but briefly refreshing you here again that while all these ions are coming, your major effort is most of them can you move them inside the mass analyzer, and that is where the pressure and the charge, these parameters are very crucial, and you need to make sure that most of the ions are actually going inside the mass analyzer, otherwise the proteome coverage will be affected. So, next let us assume that you have done a good electrospray ionization, then you are ready to separate these ions inside the mass analyzer. There could be different type of mass analyzers and different type of mass spec configuration. For example, we can have ESI Q-TOF, a popular configuration, or Orbitrap. In this case, we are going to talk about Orbitrap fusion technology, which is having a tribrid mass spectrometer, and let us have a laboratory session about using Orbitrap fusion technology and different parameters for MS and MS-MS analysis.
Once you have kept your sample in nano LC, and then you will monitor through the software. So, your sample will go through this tube, and this is first column which is pre-column where your sample will start to clean up. basically desalting, and your peptide will start to bind in this column, and then waste will go through this tube into a waste beaker. Now, once your sample will be cleaned in the pre column, then second column, which is analytical column, then your sample will start pass through this analytical column, and they will start to fractionate, and slowly the first hydrophilic peptide will start to elute, and then hydrophobic peptide will start to come. So, once your sample will start to elute from this tip of column, you can see here, and then it will start get ionized. Now, here we are applying voltage around 2.2 kilovolt, and your sample like highly charged, and they will start to elute. Now, here I am going to explain parameter for MS. So, this is MSOT. So, this is the parameter for the MS, where I kept in orbit rep resolution around 60,000, scan range 375 to 1700 MIG. So, generally for peptide it is enough, that is optimized. RF length I am keeping 60. AGC target is like automatic gain control, like how many ions you want to accumulate, so that you have to define, which I am keeping here around 4.0 e to power 5. Maximum injection time is 50 millisecond, and polarity is positive because our peptides ionize and they are positively charged. In intensity, you have to define the intensity, how much intensity threshold should be there, which is 5.0 e to power 3. Now, generally peptide is charged, so I have to define how much charge should be there, the range of charge basically. Now, here you can see 2 to 6 I am keeping. It will not take a singly charged and then more than 6, it will consider only 2 to 6 charged peptide. And then I am defining dynamic exclusion, which is 40 second here. Mass tolerance should be in ppm, and this is the parameter for the MSMS now, where I am keeping isolation window is 1.2. Now, because the MS is happening on the peptide, now those peptides have to be fragmented, and for that I am using the SCD cell high collision dissociation, and energy collision energy mode set that is a fixed, and this is the energy which I am applying for the fragmentation for our peptide. And the detector is, here is orbit rep, and the resolution for the MSMS is 15,000, and first mass which has to be detected is 100. So, these are the like parameter which I have shown you. These are the like already optimized for the level 3 quantitation. If you are using like different type of technique, if you are using a label based in case of iTrack and KMT, accordingly those parameter will be changed. So, accordingly you can do it change. So, this was the parameter for the LC and then MS parameter.
Okay, so now you are familiar that how you can use different parameters and settings for the MS and MSMS analysis. After doing a good run from these experiments from the same sample, now you will see these chromatograms, which is shown here on the screen, which shows that the time versus the intensity of these peptides. And as I mentioned, you would like to see a good Gaussian distribution of the peptides coming out of from your sample. Now, from these sample, how to make sense of this information that what these proteins are. So, if you remember that we talked briefly about looking at B and Y ions. So, again if you keep looking at walking through the entire chromatograms, you will see the pattern of the spectra, which will give you a good idea for the B ion, Y ion, various ions that are generated. And now you can use this information for the further database search. So, let us have another lab session to discuss more about these chromatograms and looking at these data.
So, till now we have seen the different liquid chromatography parameters and the mass spectrometry parameters required for the success of a LC-MS experiment. So, as you now know that the charged species enter into the mass spectrometer and get fragmented. These fragments are then detected, and by use of a suitable software, the identification of the peptide is revealed. However, just by looking at the chromatogram, one can easily deduce whether the sample run was good enough for it to be taken to further analysis. Let us now take a look at the example of a very good chromatogram. On the screen, we see a very Gaussian distribution of the peptides. This is the MS1 chromatogram. That means all the peptides which have entered into the mass spectrometer as charged species have got detected at the MS1 level. Further, based on the abundance of each of these peptides, they are fragmented at the MS2 level and detected. it is to be noted that most of the peptides which are less abundant are likely to be ignored, and only the high abundant peptides get fragmented. So, the bottom panel shows the MS2 fragmentation pattern for the selected peak. So, if you can see here, this is the MS2 pattern for the selected peak. So, you see different fragments which have been generated, and also the signal is relatively less noisy. This helps in better interpretation of the data for the software. So, we now move to another chromatogram which is not that great. So, on the screen, if you try to correlate, if you try to compare this chromatogram to the chromatogram which was previously shown, you can very clearly see that the distribution of the peptides is not a Gaussian distribution. Also, you see that there are gaps in the chromatogram which are indicative of issues with the sample and with the electrospray ionization. So, there are certain segments of the chromatogram where probably nothing entered into the mass spectrometer, or the peptides did not get ionized properly for them to be detected by the mass spectrometer. If you now look at the MS2 pattern, the MS2 here has significantly less number of fragments, which is a feature of a very bad chromatogram resulting from a very bad sample. This issue could have been due to improper handling of the sample or due to issues with the column. But this is the basic information that one can deduce from merely looking at the chromatogram at the MS1 level and the MS2 level. The raw data that is generated is then further analyzed using specific softwares which can deduce the information in the chromatogram and reveal the identity of the peptides, subsequently leading to the identification of the proteins.
I hope you got a very good glimpse of doing the mass spec based proteomics workflow, but how one could use this same set of information, these workflow for a any case study for any biological problem. In this slide, I have invited one of my PhD students, Shulina Mukherjee, to talk about how she has used this mass spectrometry based proteomics workflow in her own research. Very briefly, she will walk you through the steps and the strategies for data analysis, and in a nutshell that how it can give you some biologically meaningful insight from a clinical problem.
So now that you have an idea about the chromatogram that comes out after your mass spectrometry experiment, we will talk about how to interpret it and how to do the protein identification. So you can interpret the raw data coming out using several freely available software as well as commercial software. So one of the softwares is MaxQuant, and another software that I will be giving you a little glimpse of is Proteome Discoverer. So what the software does is it takes the raw data into account, and then it does the spectral matching and counting, and also it uses a database that's a background database. So if your sample is from human origin, you give a Homo sapiens database, and then it does the annotation of the peaks. Also, in the process of sample annotation from raw data, you can also do a grouped analysis. For example, in this case, if you are looking at a cancer sample wherein you are comparing the normal or the non-tumor samples with the tumor samples, you can give different grades of the tumor. For example, this is grade 1, this is grade 2, and this is grade 3 of a tumor, and these are the normals. So when you annotate the raw files in such a way that you already specify that which group it belongs to, the software then takes into account these considerations, and then it gives you the details of how much of the protein abundance is present across these groups, so that you can know whether you have some dysregulated proteins that can be subsequently used for identification of biomarkers. So for the setting up of the workflow, First of all, we do the database search. As you can see here, we have used Sequence HT, and also these are the steps that are followed. That is, you can annotate the parameters. That is, for example, you have put a parameter of mass tolerance which is 10 ppm. Then you have used the charge states and then the retention time. Also here you can see the spectrum files will be taken, and then it will go to the spectrum selector, then it will go to the database, and then there is another workflow which is called the percolator. Other than these things, you can also use other search engines, like you can use Mascot in parallel with Sequence HT, and then if there are unmatched spectra, it can go to the next search engine also. For example, in this case it is the spectrum confidence filter, and then furthermore you can also use other softwares for knowing into the other modifications present in the peptide. For example, if you want to know whether your peptide is glycosylated or phosphorylated, then you can use these kind of filtering to annotate those changes in your peptide. So how does the data look? So as you can see, after going through a lot of filtering criteria, the data that comes out is of high confidence, because you have put on stringent filters so that whatever hits you get are the true data and not false positives. So here you can see that there are different tabs associated here. For example, this is where you have annotated whether the protein FDR confidence, how much is the level. So here you can see all of these proteins that have been identified have high FDR. So false discovery rate is a statistical value that estimates the number of false positive identifications among the peptide, and it is also a measure of certainty for the identification, as in how much you are confident that the protein you have identified is a true match. Then we also have a contaminant database which we can plug in the workflow. The contaminant databases will actually indicate presence of keratin or serum albumin, which are high abundant protein and often are responsible for giving false positives or masking your actual protein of interest. So as you can see here also in our data, we have got serum albumin, and the software has marked here as true. So while all the other proteins there is false, in case of serum albumin it is marked as true. So you can remove this kind of protein in case of your subsequent analysis. Other than that, we will also have a plethora of information like the unique proteins. So the unique proteins again gives you an idea as to how confident you are of identifying the particular protein that has been ascribed to. For example, the first hit, which is a neuroblast differentiation protein, has 370 unique peptides. That means the mass spectrometer has encountered the peptide 370 times, or it has actually annotated the protein, the software has annotated the protein with very high confidence. Then you can also know about the score, which is the score that is given to by the search engine, and many other details. So thus you will have all the information available after you have run the protein through, after you have run your raw file through a software, and the software has different tabs that you can customize, and you can set your parameters that you are looking for. So basically we have started from here, that is we have taken a cancer sample and normal samples, we have used a patient cohort, then we have extracted the protein, and then we have run the samples in the mass spectrometer. So after doing all these exercise, you have got raw values, you have got different peaks that you need to know that what are they. So you have used a software which I told you that you can either use a freely available software like Maxquant or a licensed software, whichever resource is available to you, and then you can use the software and the various parameters to now do the data mining. So after the data mining, what you are actually looking for is something like this, wherein you can see a clear difference between condition A and condition B. So as you can see, there are a signature list of proteins which are highly abundant in condition A, and there are a signature list of proteins which are highly abundant in condition B. So these are indicative of the actual biological changes that are happening in the patient sample. Furthermore, you can do the data curation and network analysis using doing several bioinformatics software like StringDB, MetaboAnalyst, etc. And now you can see that after doing the whole exercise of using mass spectrometer and the software for annotating the data, we can map the proteins in various networks like this, and then we can also see which are the ones which are classifying the different grades of tumor or are different between the control and the cancer samples. For example, these are a set of markers, as you can see. So you can see in this one, this is very high in the C sample, which is the control sample, and relatively low in the grade 2 samples. Similarly, there is a reverse trend for this protein. You can see QO4637. So this is again showing a sequential increase, that is, it is low in the control samples and going high as the tumor progresses. So thus, using these kind of tools, you can answer a lot of biological questions.
Alright, so we started with a workflow of mass spectrometry based proteomics. We talked about how to do the protein quantification, protein digestion, again we have talked about peptide quantification. Then you are ready for doing the LC-MS-MS based analysis, so liquid chromatography and MS parameters, what we generate, the chromatograms, how to interpret chromatograms, and how to review the whole data set, make more meaningful insight from this data for the clinical case studies. I hope this gives you very basic, of course it is not so detailed, but a good glimpse of the workflow involved in doing MS based proteomics. A lot can be done using mass spectrometry based proteomics. We have just talked about protein identification and label free quantification workflows. You can also think about quantitative proteomics, which I talked in the theory classes earlier about using iTrack or TMTs, and those workflows can be very useful as well. But as long as you have done this workflow of sample preparation and their separation very well, then you are ready for the quantitative proteomics based workflow as well. Thank you.